Hi everyone, and thanks for tuning in to BEaTS Research Radio. My name is Hallie Arnott. I'm a biomedical engineering master's student at the University of Ottawa, and I'll be your host on today's episode. As you might be aware, we're normally streaming from a lab at the University of Ottawa Heart Institute, but for now we're using our online platforms to help continue communicate scientific research to the community. Our guest today is Dr. Baptiste Lacoste. Dr. Lacoste is a scientist at the at the Ottawa Hospital Research Institute and an assistant professor at the University of Ottawa in the Faculty of Medicine's Brain and Mind Research Institute. His research is centered around studying neurovasculature interactions to gain a better understanding of brain and mind health. Dr. Lacoste started his research career in neuroscience at the University of Bordeaux in France before completing his doctorate at the University of Montreal. He then completed a postdoc fellowship at the University of Montreal Neurological Institute at McGill, and continuing his passion for neuroscience, he completed a second postdoc at Harvard Medical School. Today, he is here with us to speak about his research advancements in the field of neurovasculature. So thank you very much, Dr. Lacoste, for joining us today.
Thank you for having me. It's a pleasure.
So before we get into some technical neurovascular questions, I'd like to first ask, what got you interested in neuroscience, and what do you find the most fascinating about the field?
Great and difficult question. It goes a long way. So I think the first thing when I was a young student in France, this is where I come from, as you will guess from my accent, I guess, it's a course of biology by a very good professor who introduced us to neuroscience with all the synaptic communication system, all this, transmitter release and the effect on the receptor and postsynaptic communication, how these neurons communicate together, and how magical it is because it brings you all those feeling, this consciousness, this sensory stimuli, and all the stuff that actually makes us human. So I think that's where my passion for neuroscience really started, pretty early on.
Mm-hmm. Yes, that's definitely very interesting. And what made you, was that in your undergraduate that kind of got you interested, or was it more towards doing graduate studies?
Right. It was like a long time ago. So it was a high school teacher actually, who a biology teacher, which was a great person for opening the students' mind. And I remember she was inviting people from this institute in France, the CNRS, so a research institute in France. And then those people would come to the classroom and talk about what they're doing. And I remember exactly that they were talking about, those G protein-coupled receptor trafficking in the membrane. And I was like fascinated by all this physiology, these pathways that lead to, behavior. And so that definitely, was the spark. And then I continue on. And I wanted to be a medical doctor, to do like daddy. I finally decided that it was not made for me. I prefer to work with mice than humans.
Yes. And as you were just saying there, it's the circuitry that's incredibly interesting, and we hear a lot about specific neurons and the electrophysiology. But would you mind explaining to us a little bit about the importance of studying neurovasculature?
Yeah, that's okay. So let's start by saying the classic thing is, of course, the brain is made of billions and millions of neurons, but if you talk to people interested in glia, for instance, they're going to tell you that you have much more astrocytes in microglia, for instance. So that's the complexity of the brain is, how many cells and cell types it's made of is often forgotten by people. But the cell type we are the system we are studying is the vascular system, made of little parasites, endothelial cells, many things. But to make things simple, I would say that if neurons don't get the energy supply, they cannot work. So it's very simple. If you do an electrophysiological experiment in your artificial medium or CSF, or you're going to add oxygen, you're going to add glucose for those neurons to function. So in the brain, the only way you can bring those nutrients is through the blood stream. So that's the importance of the vascular system and studying it, because if there is an issue in that specific system, whether it is during development or after an injury or during a long process of aging, it leads to neuronal dysfunction. So that's something that is very, very much studied in the case of vascular cognitive impairment, for instance. You might have heard of vascular dementia, which is a form of dementia. Alzheimer's disease is more and more recognized as I would say, a vascular disease of the brain, because one of the first things that happens is a vascular dysfunction. So that's why also I've found during my first postdoc at McGill, where I discovered this vascular system studying Alzheimer's disease. I found it fascinating that people were not actually studying it enough and not even properly. So I think that draws me into that stream. And for my own research, so I continued with the lab of Shenhua Gu at Harvard Medical School, where I studied how those vessels develop. So with that background of postdoctoral fellowships, where I studied dysfunction of blood vessels, but also the genetics, which are driving development of blood vessels, I came to open my own lab and started to study neurodevelopmental disorders and their impact on vascular systems of the brain.
Right. So your research right now is kind of giving a broader spectrum to what is maybe more commonly studied in just the neuronal circuitry. So in your lab, do you kind of study the whole range? So from the vasculature all the way to maybe electrophysiology, or is it mostly focused on that vasculature part?
So that's an excellent question. So what we do, and we cannot do everything ourselves. That's also why on the publication we are talking about today, there are 25 authors. So I would define my lab as a neurovascular lab, where we do a lot of vascular biology. So definitely we do a lot of cell biology, genetics, but also we move to mouse behavior, and we have some, I would say, simple, basic electrophysiological readouts, such as LFP measurements. So, but we are not doing everything by ourselves. We did collaborate with a lot of people in order to get help on the neuronal side of things, on the genetic side, on the interpretation of the data, etc. So our lab is doing a lot of things. Thank you, my students, to bear with me. But we need the environment of the Brain and Mind Institute, of the OHRI neuroscience program, of the Faculty of Medicine. It's actually a very good place to be doing some neurovascular research, definitively.
Mm-hmm. You're absolutely correct. The collaborations are extremely important as you become more in depth in a research field. You realize you need the chemists, you need the physicists, you need everything. So it's the biologists, everybody's involved, and that's when you see these incredible publications like the one that your laboratory was a part of. And I understand that your research team and the collaborators published, I think it was the beginning of this month, and in Nature Neuroscience, and it was on the topic of vascular development in autism. So do you mind now, let's get into that topic, and let's speak a bit about that publication. What kind of sparked this publication, and what were your findings?
Okay, so let's maybe start by the very beginning. When I was at Harvard in my second postdoc, I was already thinking about the lack of studies on deficits in neurovascular systems and neurodevelopmental disorders. I was already kind of frustrated by the fact that everybody is studying. I mean, I love my Alzheimer's colleagues, but and other diseases, but I was finding this field very competitive and also very crowded. So I looked around, and I was very surprised that the field in general of neurodevelopmental disorders, which includes Down syndrome, Fragile X syndrome, autism spectrum disorders at large, was not connected to the neurovascular systems. And thinking about it, I was diving into more literature. At the end of my postdoc, at the time I was transitioning to my own lab, and I found this paper showing a paper I was afraid of at the beginning. I was like, oh, shoot, somebody has done it. But actually, not quite. So it was a post-mortem studies, so people taking kids from 8 to 12 years old who passed away but were autistic as well, and they looked at the brain vasculature. And one study only, said that those vessels looked different. They probably were a little bit more tortuous and definitely different from a normal brain. But that was the paper. That was like the observation in postmortem fixed tissue, which I have a lot of respect for, but it was not a kind of mechanistic, first of all, study. And then the causation between vascular deficit and autism was not established. So that's why I decided to dive in. So we wrote a grant. We did a lot of work to get preliminary data. And 1 year and a half after starting the lab, we got funding from CIHR, and we could really take off with this adventure.
So what were the, so once you started working and got the grant and started working on this topic, was that paper that you were speaking about, was that the only one out into the world at the time? Was there more kind of?
Yeah, there was like getting more and more. So, and the more you read, the more you discover that you are ignorant. And we were discovering on the go that we missed a lot of things. So, for instance, we found some published studies a little bit even earlier than the paper I was mentioning about this postmortem study in brain imaging in humans. And people using magnetic resonance imaging, or MRI, were finding, I'm talking about a paper in 2015, some differences, abnormalities in the way the brain seems to be perfused. So when I saw that, I was like, oh, guys, so we are in the right direction. And that's basically what we say in the introduction of this paper. The way we set up the hypothesis is based on that postmortem study, but also on those imaging studies in humans. So the data from those papers was saying there are regions of the brain that seems to be hypoperfused, but other that seems to be hyperperfused. So to me, this is like a perfect excuse to say we are going to look at neurovascular coupling. But not only that, because it's a neurodevelopmental disorder, we're going to look earlier in development and see whether those deficits that might be observed and confirmed in mouse models might result from an earlier mechanism or that can either drive vascular deficits per se, but also those vascular deficits might drive neuronal deficits on the long term. So that's basically what the paper is about.
Right. And were the findings from the paper, were they what you thought, or were they a little bit different? Was there anything that kind of came up as surprising?
Yeah, so like every science lab, I think, let's be honest, you take one direction, you find something and you shift, you take the rail and you change direction. So we kept on changing directions. But the good thing about, the very surprising thing about our study was as much as we were adding some data, some findings from either our lab or collaborators, they will all converge in the same conclusion. So definitely we would at the beginning say, it might be the regulation of metabolism. Oh, it's not. No, it might be the regulation of this and this and this and this. But we really found that in the young brain of those mice, the primary event that we could identify with our tools was a defective angiogenesis. So what was this inability of young vessels to develop a complex network of blood vessels in the brain? So that was kind of the first surprise, because we would expect to find something about angiogenesis because of this postmortem study, but we were surprised to find how strong the phenotype was, how consistent between mice it was. So that was the surprise. Then we moved on and we looked at functional readouts, which were either blood flow measurements in mice or electrophysiological measurements in mice, and then behavior in mice, and vice versa, where we can talk about this a little bit later. We could induce that specific mutation we're talking about exclusively in blood vessels using a conditional genetic approach. And we would find that this conditional vascular mutation would drive neuronal phenotypes and behaviors. So that was the big surprise. I even said to my student Julie, who was the driver of this work, when she was hired in the lab, she said, you're going to find something. You take a mouse which has this huge deletion, we can talk about it, this 16p11.2 deletion, you are going to find something, because it's going to impair development of cells, that's for sure. But to find that many things and knowing that all those findings, even independently obtained by different teams, were all converging towards this is an angiogenesis defect which on the long term drives endothelial dysfunction and neuronal abnormal development. That was the big surprise, and a very good surprise.
Yes, definitely, and that's great that you know, the research is headed in the right direction for becoming very translational. Because there's one thing to find something in the lab, and then there's another to have that become translated into the clinic. So my next question is, right now autism is mostly, it's just I think from my knowledge, behavioral studies are used to kind of have kind of a spectrum to decide whether or not, you know, somebody is autistic or not, and it's these behavioral studies, and it's not very or most of it isn't very physiological. Mm-hmm. So do you think that your findings from the study and being able to see these physiological differences, mm-hmm. do you think that could be translated into the lab and be used by doctors to determine the spectrum of autism?
Yeah, so that's a very good comment and question. I think so if you really consider how broad research on autism is, you definitely have behavioral studies. So in humans, I would say you have behavior diagnostic, which is the main thing to determine where on the spectrum you are. But you also have imaging studies. So by brain imaging combined with behavior, you have kind of the physiology, if you want. And then I think people are going to this physiology, but on the neuronal side of things. So that's also why I think what we can bring is maybe two things. Is as you might have seen, as readers of the paper would see, we are not bringing a solution. We are not telling you we found a treatment for autism. We are not even saying autism is a vascular disease. That's not what we're saying. We're saying that, first of all, vascular deficits play a role, and that's causally linked to pathogenesis of autism. So that's the first conclusion. The second one is, since people use a lot of behaviors and physiology on the neural side of things, maybe that vascular dysfunction observed in autism, first of all, it would be nice to really confirm it in humans. That's what we are trying to do, and convince people to do imaging with and for us. But also consider it as a diagnostic tool. So, for instance, if you know how or to what extent or where perfusion of the brain in autism might be affected, you might turn it into detection of autism or part of this diagnostic card when you have to check many things. So that's one thing. The last thing I would say, it's opening new doors, and it's opening new doors for treatments. We are not proposing a new treatment. But if you know that, for instance, as the paper concludes, the endothelial cell, which is the cell forming the brick of the tube of the vessel, has a deficit that is specifically about forming new branches and regulating blood flow, for which we have a lot of avenues, because we also screen all the genes and we have, targets. Maybe then people can, and including us, can follow up on finding drugs that regulate and rescue those deficits. So that's the translatability of the study, but we are not there at all. But I think opening a new door, saying, "Hey guys, vessels play a role in autism," was already a big finding.
Right, definitely. That's absolutely lots of great comments there about, just there's lots of different ways studies can be translated, and seeing different avenues is great. So my final question about this paper is, what are the motivation, or has the study motivated a next study? What's coming next from yourself and the collaborators on this project?
I won't say. No, I'm just kidding. So the thing is, this paper is nice. It finds something cool, but the issue with this is it's opening a lot of possibilities. So we're gonna have to choose and focus. So definitely, also thanks to new fundings coming in recently, we are going to study how the brain, in response to these vascular deficits, shifts its metabolism. So on the track of basic science, fundamental findings, for which I must admit I was trained for, rather than translational studies, but I want to, and we want to understand how the brain adapts to those vascular deficits. So we have very good findings already in the pipeline, showing that definitely the way the brain uses glucose, for instance, is much changed. And we're going to dive into and try to find an explanation. The other big area is, of course, trying to rescue those deficits. So we have the next big project is going to be testing drugs or screening drugs, candidates that either connect to those genetic changes we found, or drugs that are known to regulate those deficits that we found, because drugs already exist and can be repurposed. So that's I would say the two main avenues we're gonna pursue.
Right. That's yes, that's great. And my next question, it kind of ties in still to this topic, but in the context of these pharmaceutical interventions that you're talking about for neurological disease or conditions, the treatments available and the therapeutics available for neurological diseases and conditions, there seem to be few and far between, most often for the neurodegenerative diseases. Mm-hmm. Why, compared to other diseases where this seems to not be as big of an obstacle, why do you think that we haven't really gotten there in terms of therapies for neuro diseases and conditions?
That's a difficult question to answer. I'm gonna just say my grain of salt. I think most of the issues come from experimental design. I think most is about what you define as a good model for a specific disease. It's not because you find a drug that rescues a behavior in that mouse, which I did during my postdoc at McGill, that you found a drug to cure Alzheimer's disease, for instance. So it's such a multifactorial, I'm just taking this example because this is kind of the king of neurodegenerative disease, but it's such multifactorial disorders that having either a single hit, drug or a multiple system targeting, we're not there yet. It's extremely complex to dissect those mechanisms. The parallel between our study in autism and Alzheimer's is not easy to make, because Alzheimer's disease has a different set of vascular deficits, including a breakdown of the blood-brain barrier, which is the wall of the blood vessel. So it's a little bit more complex. But I think in terms of how endothelial deficits and this neurovascular unit, which is the ensembles of whatever is the tube and what's around, have some overlaps with neurodegenerative disease, which is very interesting. But the reason why we didn't find cures, it's just, I think, because we don't have yet the right design models and probably the right standardization of the procedures and the way people do science. So I think it's I don't want to turn into politics and criticisms, but I think it's just so complex that it's even utopic to think that we might find an easy road for treatment for these things. That's not just my point of view, I would say.
Yes, definitely. And trying to replicate these diseases outside and conditions outside of the body it's very hard, and even understanding how they work inside the body is already a difficult task.
But I must add something, just for the credit of all of us scientists. I think we have to think as a community, and each one is going to bring his own little brick to the big wall. I think it's ambitious to think that drugs can be found pretty fast and easily, I think, but we as a community, we just put the little piece of the puzzle, and absolutely. One day it's going to be some.
Mm-hmm. Yes, and as you're saying, the collaboration is extremely important, and the little bricks that come, they come in little pieces, and something that somebody else knows that somebody else doesn't comes together, and that's the beauty of science, and you need a lot of different people, a lot of different expertise, ideas to come to some conclusions. And that paper that your group and your collaborators have published is I think, a great result of that collaboration. So I think that concludes our interview today. Once again, I'd like to thank our guest, Dr. Baptiste Lacoste, for joining us on our program and for speaking to us about all of the advances in neuroscience and the importance of brain and mind health and neurovasculature. The BEaTS Research Radio is supported by the University of Ottawa Heart Institute, the BEaTS Research Lab, and the Department of Biochemistry, Microbiology, and Immunology at the Faculty of Medicine at the University of Ottawa. Our AV producer is Alex Ross, and our director is Dr. Emilio Alarcon. Thank you for tuning in, and don't forget to subscribe to our YouTube and podcast channels. We'll see you again soon for some more science in our upcoming episodes.